Hello and welcome everybody. I am Ania Lichtarowicz, your moderator for today's Frontiers in Science Deep Dive. It's fantastic to see viewers from over 64 countries joining us. Whatever the time zone, you're in good company. It is great to have you along. If you're new to Frontiers in Science, this is our flagship multidisciplinary open access journal at Frontiers. We publish exceptional peer-review-led articles on transformational science, key to accelerating solutions to the global challenges facing all of us and our planet. We publish these articles across unique hubs with content to suit all kinds of audiences, extending the journal's reach and impact for audiences engaged with science, policy, and society. To date, we've published 33 hubs, spanning 122 articles that cover a diverse spectrum of transformational science. The research has been cited over 1,400 times, viewed over 4.1 million times, and is regularly picked up by the media. Today's deep dive focuses on this lead article titled Waste to Value: Microbial Electrochemical Technologies for Sustainable Water, Material and Energy Cycles, led by Professor Uwe Schroeder, and we are proud to give the article and its hub a home in Frontiers in Science. Every year we produce enough wastewater to fill Lake Geneva 4 times over, with half of global wastewater discarded and the rest expensively and inefficiently treated for reuse. Emerging microbially powered tech could reclaim these resources from the drain, save money and reduce environmental harms, presenting a transformative approach to wastewater treatment by simultaneously enabling energy recovery, clean water production and nutrient recycling. In their Frontiers in Science article, the authors review these microbial electrochemical technologies, METs, examining how they can convert organic waste streams into electricity, fuels, fertilizers, and usable water more efficiently. The article highlights how this approach to wastewater treatment can support energy access, environmental health, and decentralized sanitation in underserved regions. Pilot deployments from the UK's Glastonbury Festival to field trials in Uganda, Kenya and South Africa are demonstrating its potential to reclaim energy and harness the 359 billion cubic metres of wastewater being flushed away annually. Stay with us and then join the conversation as we explore the scientific, engineering and regulatory challenges that must be addressed to scale these technologies and discuss the need to convene a coalition of stakeholders, including researchers, water providers and policymakers, to transition from pilots to large-scale implementation. This is the Article Hub. Alongside the leader article, we also have an editorial by Professor Bruce Rittman, who joins us today, and Professor Cesar Torres from Arizona State University, USA. A viewpoint article led by Professor Vera Goudet from Purdue University Northwest, also in the USA. And a policy outlook by Professor Mansour Kadir from the United Nations University Institute for Water, Environment and Health in Canada. The hub includes a lay explainer, a video and a version of the lead article in Frontiers Young Minds, our science journal for kids. This is today's schedule. First, we will hear from the authors of the lead article, Professor Uwe Schroeder, Dr. Liz Heidrich, Professor Falk Harnisch, and Dr. Deepak Pant. Then, joining us for the panel discussion, we will welcome Professor Bruce Rittman, author of the editorial on the lead article. First, I'm delighted to welcome Professor Uwe Schroeder, Professor of Electrobiochemistry at the University of Greifswald, Germany. A leading professor of biochemistry and electrobiochemistry, he explores microbial electrochemical technologies, green chemistry, and microbial fuel cells, focusing on turning waste streams into useful resources through bioelectrochemical systems that can support cleaner water, materials, and energy cycles. Uwe has contributed fundamental insights into extracellular electron transfer and the design of bioelectrochemical reactors, helping bridge the gap between laboratory discovery and real-world application. And this played a central role in shaping the field through his interdisciplinary collaboration and previous roles, such as Executive Director of the Institute of Environmental and Sustainable Chemistry at the Technical University of Braunschweig. He's also Field Chief Editor for Frontiers in Energy Research. Uwe, thank you again for bringing this article to Frontiers in Science. The floor is yours.
I would like to first to welcome everyone also from our side, and I would like to start with this presentation with a simple question to understand how and why microbial electrochemical technologies work. And it actually comes down to two questions concerning our own respiratory metabolism. And that is the question, why do we eat? And also, why do we breathe? This is actually quite easy to answer. We need, of course, the food as a source for chemical energy that we need for our survival, and we need the oxygen to burn the fuel and to create energy. And this is why we also say that we burn calories. But it's not, of course, a simple combustion as we would use it in a power plant quite inefficiently, one has to say. Biology has developed much more elegant ways, and this, of course, concerns the respiratory chain, in which the electrons are transported basically from the oxidation of the fuel or of the substrate along the respiratory chain to the oxygen. Along that way, they do work, produce ATP, and at the end, they are transferred to oxygen as it is called the terminal electron acceptor. And this happens actually within every cell in our body, and it also happens in the entire aerobic world. And if we don't have organics, if we don't eat, we starve. If we can't breathe, we suffocate. Both are basically parts of the same equation. Of course, not the entire world is aerobic, and there are vast compartments in which there is not enough or no oxygen at all is available. And of course, here evolution has developed quite interesting pathways to still allow microbes to live and also to respire. And one very interesting pathway is, instead of taking up soluble electron acceptors into the cell, is to dispose electrons across the cell membrane to an external electron acceptor, as it would be like a mineral like iron oxide or humic compounds. And that was discovered only at the end of last century. And at that point, it was considered a scientific peculiarity. And it has become quite obvious over the past decades now that this kind of process takes place all over all kinds of anaerobic compartments. It takes place under basically and virtually under our feet and in soils and sediments and so on. And it is interesting from the perspective that this kind of discharge of electrons across the cell membrane, this is something that we call extracellular electron transfer, is the key to what we nowadays refer to as microbial electrochemical technologies. The reason is that we can now replace the iron oxide or the mineral in the soil by an electrode. And now we kind of create a win-win situation, because on the one hand, we allow the microorganisms to dump the electrons from their oxidative metabolism towards the electrode. And for us, it is a win situation because we can gain the electrons in the form of an electric current, which is the reason why these kind of bacteria are often referred to as electrogenic bacteria. The microbes do this kind of electron delivery on a kind of very on a willing level. They grow nicely and form thick biofilm layers on the electrodes because it gives them the chance um, to survive in an environment in which usually they would not have a chance to survive. And that allows us to take benefit from these kind of, I would call them electrode respiration processes, because it would now allow us to combine like the degradation of organic matter with the generation of electricity. And that in doing so, it would allow us to exploit bacteria as living as biological electrocatalysts. That is something that is really interesting because it would mean that we don't have to artificially cast an electrocatalyst on a surface, but the catalyst, the cells would grow themselves, they would replicate, they could repair themselves, or the biofilm, and would allow a long-term operation. And the bacteria are also usually, and the whole food webs are adapted to quite complex substrates for which we would not have any chemical or artificial electrocatalyst for this kind of conversion. And the microorganisms are also adapted to feed on dilute substrates, which means low concentration, organic substrates at a low concentration level. And that is quite interesting because as a society, we don't actually have technologies to deal with low value resources. And that is something that is, when I say low value resource and put that in relation to a high value resource that we are usually or which we have the technologies, I can show this kind of a comparison. Like gasoline, this is a typical high value energy resource. It contains 8 kilowatt hours of chemical energy per liter, versus wastewater typically contains more or less about 2 watt hours per liter, which means we have something like 4,000-fold more energy in gasoline as in wastewater. Which, of course, leaves the question open why we should actually deal with this kind of low-value resource. And at that point, I'd like to hand over to Anya and to Liz again.
Thank you, Uwe, for those insights and setting of the scene for today's discussion. Our next speaker is Dr. Liz Heidrich, lecturer in environmental engineering and director of research for the School of Engineering at Newcastle University in the United Kingdom. Liz is a leading researcher in microbial electrochemistry, developing bioelectrochemical systems that enable the direct conversion of organic matter into electric current, and vice versa. She has led international efforts to advance these technologies from laboratory research to pilot-scale deployment under real-world conditions, and her Met Zero project, which aims to support the water sector's transition to net zero using microbial electrochemical technologies, has secured a £1.2 million Engineering and Physical Sciences Research Council Award. Her research is driven by the ambition to better understand and ultimately harness microbial communities for sustainable engineering applications. Welcome, Liz. The floor is yours.
Thank you, Anya. Yes, welcome everybody. So, as Uwe talked about this, what is the scale of this low-value resource that we are discussing? And on an individual level, each person produces approximately 120 to 150 litres of wastewater per day. Obviously, that's an average value. In different parts of the world, this number might be vastly different, but it gives us a reasonable working value. And that means that a household of 4 will produce about 500 to 600 litres per day, or half a metre cubed. And that is a reasonable volume, but actually, when we look at this globally, obviously that's intrinsically linked to the global population. And in the last century, we've seen huge population rises and therefore huge rises in the amount of this wastewater produced. And likewise, kind of moving forward, this will increase as the population increases. Further factor in this is that as it currently stands, there's a large proportion of the world that actually isn't connected to safely managed sanitation. And therefore we have this challenge of collecting all of this wastewater and to enable us to try and treat it and recover something from it. So what is wastewater? It's not simply what we flush down the toilet. At a household level, it's both what we kind of flush down any drain, it's our washing liquids, it's our shower water. In some countries, the wastewater system is also linked to drainage. So a water event would lead to a huge spike in the amount of water that was in those sewers and drains. It's also all the effluent from shops, from hospitals, from schools, from all of that urban network that surrounds us. And it's the wastewater that's generated by industry and agriculture. And this can be very different in very different places. As we see across the bottom of the screen here, there's huge engineering challenges in gathering, collecting and treating all this wastewater from these different places. So the following table kind of collates that data, and we can see at the high level, globally, at huge volume, about 316 times 10 to the power 9 meters cubed per year is produced, vast quantities. About 60% of this is collected, only around half of it is treated, and just about 11% is reused. That reuse value is largely, at the moment, reusing the water itself. So removing, through treatment, the pollutants and reusing that water, usually in agriculture, though in some countries it may be actually reused for potable water. What if we reused it in a different way, though? What if we didn't simply remove and discard those pollutants? What if we saw those pollutants themselves as a resource that might be reused? And the first and most obvious resource that Uwe alluded to is this organic energy that is contained within the wastewater. And he showed that picture of the delicious beef burger at the start of this slide deck. But actually, to bacteria, the waste that we humans produce is a delicious bacteria. It is a delicious burger. So that waste water and the organic load that is in it is food and calories for bacteria, and therefore it has a calorific or an energy value. And if we take kind of basic domestic wastewater, about a litre of that might contain 8 kilojoules of energy, which means in a meters cubed, which is about double what we said a household might produce, there would be about 2.2 kilowatts of energy, enough to power a heater for about an hour. So we are not talking about enough energy to power homes, to power factories, to power anything. We're talking about a very low level energy source, but nonetheless, there is energy in it. If we extrapolate that energy up and look at the big numbers globally, of course, that energy is produced wherever there's population, and therefore it multiplies up. And globally, if we were to collect, gather, and get all that energy from the wastewater, it will be about 100 power plants equivalent. Clearly, that actually wouldn't happen. We'd never be able to recover all of the energy, but that's just numbers to show the kind of scale of the energy that's in there. Possibly the more important and other half of the story is that currently the wastewater that we treat is done through a very energetically expensive process. We've got these lovely images here on screen of oxygen being bubbled up through tanks of wastewater. And this is how wastewater is treated. We bubble the oxygen through so that bacteria can breathe the oxygen, eat the food, and then therefore treat the wastewater. The energy required for treatment is about a quarter of what is contained in that wastewater. If we could do this a different way, if we could get the bacteria to breathe on something different, and Uwe talked about those different breathing cycles, then we could save this energy, equating to about 22 power stations. And it's not just energy. Wastewater contains nitrogen, a really key and important nutrient in our society and in our agricultural systems. If we were to collect it from the wastewater, it would meet about 11% of the current global demand for ammonia. And of course, in doing that, it would offset some of the energy from the really expensive, energetically expensive process that ammonia is currently made from. Phosphorus or phosphate too is also present in wastewater. And if we were to collect all of this, we could meet about 7% of the global annual demand. And if we could gather those nutrients, if we could gather the energy, if we could remove all the pollutants that was in the wastewater, we have, of course, got the water itself, which in many parts of the world, where it doesn't rain quite as much as Newcastle, that water is extremely valuable. So coming to the end of my slide, I kind of go back to the really basic water cycle. All of us learnt this in primary school, and this water cycle has been in operation in the world since time began. Man's interaction with this water cycle has not been cyclical to date. It's been very linear. We abstract water from the environment, we treat it to meet our standards, we sometimes store it, we distribute it, we use it, and in using it, we therefore pollute it, and then we dispose of it, and it's sent back out into the environment. What the authors of this paper, and many people on the call, would argue is that actually we need to be more cyclical with this. We need to create more of a circularity in the way that we use the resources that we have. And I'll pass over now to my colleagues who will talk about how METs may play a role in increasing the circularity of this water system. Thank you.
Thank you, Liz, for those insights into the scale of the challenge involved and the opportunities for this technology. It always astounds me how much wastewater we produce every day. Our next speaker is Professor Falk Harnisch, co-head of the Department of Microbial Biotechnology at Helmholtz Center for Environmental Research, Germany, and Professor for Electrobiotechnology at Leipzig University. Falk is an expert on microbial bioelectrocatalysis and bioelectrotechnology, and his research focuses on developing bioelectrotechnological processes that combine microbiology and electrochemistry to transform wastewater and other side streams into energy, chemicals, and sustainable fuels. He has helped shape the field of bioelectrical chemical systems, driving innovation at the interface of microbiology, electrochemistry, and engineering, and launched the first book on bioelectrosynthesis. He is a fellow and past president of the International Society for Microbial Electrochemistry and Technology. Welcome, Falk, and over to you.
Thank you, Anya. It's a pleasure being here with all of you, and thank you, Uwe, for setting up the fundament and Liz telling us why we should do it. And I'm now happy to introduce a little bit the basics, how we should extract energy and nutrients from wastewater using microbial electrochemical technologies. And I'm happy to start with the microbial fuel cell. So, and I will walk you now through this diagram stepwise here from the left side to the right side. So we start on the lower left button with the wastewater. The wastewater enters an anaerobic chamber. Anaerobic means oxygen-free, where we have this aforementioned electroactive microorganisms that were introduced by Uwe. And these microorganisms then oxidize the organic compounds, so the equivalent of COD that Liz also referred to, into carbon dioxide. And the electrons released thereby are transferred by the microorganisms here in purple to the electrode, which is an anode because it accepts the electrons. The electrons are here shown in green on the picture. And there are different ways of the microorganisms to perform this so-called extracellular electron transfer. We will not go too much into detail of this now, but in principle, there are some that require physical contact and some that don't require physical contact of the microorganism with the electrode. And then the wastewater can be released from the anode chamber as treated wastewater, and most importantly, the chemical oxygen demand, so the organic load that is therein, is decreased. However, we have to follow the path of the electrons now here in green. So the electrons travel from the anode via the external electrical circuit to another electrode, which we call cathode. And this cathode, for a microbial fuel cell, is inserted into an aerobic environment. So finally, it's a liquid that is actively pumped with air or purged with oxygen. And at the cathode, the so-called oxygen reduction reaction happens, meaning, like in our respiration, oxygen is reduced. It also needs 4 protons for this, as you can see here, and water is formed. And by doing so, the entire cycle is closed. Electrons are intercepted at the anode from microorganisms that gain it by oxidation of organic compounds, and then finally transferred to oxygen as well, like in aerobic respiration. But we can harvest part of this electric energy, as depicted here by the light bulb, for using it. But we have to be aware that there are not only electrons traveling. It's also important, and this is especially for our last part of the of this section, that also ions have to be transferred. And for every electron that moves from an anode to a cathode, a cation, so a positively charged ion, has to move from the anode compartment to the cathode compartment, or vice versa, a positively charged cation from the anode, from the cathode to the anode side, or a negatively charged anion from the anode to the cathode side, if I may have said this wrongly a minute ago. And this is the so-called charge balancing ion transfer that has to take place between anode and cathode compartment. And by moving forward to the second type of microbial electrochemical technology, we now see the microbial electrolysis cells. And the good thing is now we can almost skip the anode side, because it's almost identical to what we have just seen for a microbial fuel cell. The main difference is now that on the cathode, where before oxygen was reduced, now protons are reduced to hydrogen, which can happen without the help of microorganisms and with the help of microorganisms, so abiotically and biotically. Or, for instance, when we make methane, carbon dioxide and protons are used for making CH4, which is methane, but therefore we need microorganisms as electrocatalysts that facilitate this chemical reaction. And as you can see also from the color coding of the chambers, here both the anode and the cathode compartment are anaerobic. And we can now move even further and have a look on the energetics. And in very brief, it means that in a microbial fuel cell, the microorganisms extract a certain share of the chemical energy in their substrate, so in the chemical oxygen demand from the wastewater perspective, and use it for their living. The most of the energy by the extracellular electron transfer is transduced to the anode and can be harvested as electric energy. So finally, we can transform chemical energy into electric power. When we now look on the microbial electrolysis cells, the picture changes a little bit, because for driving the hydrogen evolution reaction on the cathode, for instance, that was just introduced, we need additional electric power to make the entire device running. And you now may ask, yeah, we can do this also with conventional water electrolysis, meaning we have a just conventional abiotic anode reaction where we make oxygen on the anode, and we also make hydrogen at the cathode. So we get the same product, hydrogen, and an additional product, oxygen gas here, but one has to know that by a rough estimation, the energy that is needed per mole of hydrogen that is formed in a microbial electrolysis cell is by more than factor 4 lower than for conventional water electrolysis. So we need less energy in this case. And finally, we come to the maybe in this context now most complex, but also I think one of the most appealing ways of using microbial electrochemical technologies. And I will also walk you through this. So far, we have only focused on the carbon and not on the nitrogen that is also contained in the wastewater. When we now have an ammonium rich wastewater, so still dilute compared to industrial streams, but compared to other wastewaters, we have a lot of ammonium therein. It also enters the anode chamber of a microbial electrochemical device. Still, the microorganisms oxidize the COD, so the organic carbon, and make carbon dioxide. The electrons travel from anode to cathode. But as already introduced, we also need a charge balancing ion transfer from anode to cathode compartment. And if we have a comparable high concentration of ammonium, then the ammonium travels from the anode as a cation to the cathode to assure the charge balancing ion transfer. And during operation, a pH gradient is formed because protons are released on the anode side, meaning we have a lower pH, whereas protons are consumed on the cathode side, meaning the pH increases. And if the pH increases more than 10, which we can do by engineering, the ammonium is transferred to ammonia. And ammonia, NH3, is a gas that easily leaves the liquid and can be harvested by an established technology, for instance, membrane stripping. And this already creates a link to established wastewater treatment technologies. And I'm happy to hand now over to my fellow colleague, Deepak, after his introduction.
Thank you very much, Falk, for that. Last but not least, it is my pleasure to introduce Dr. Deepak Pant, Senior Scientist and Project Manager at the Flemish Institute for Technological Research in Belgium. Deepak's research focuses on advancing sustainable bioelectrochemical systems for resource recovery and carbon utilization, and centers on electrosynthesis and the development of innovative electrochemical systems for carbon dioxide conversion and microbial processes. Together with his team, Deepak has developed and upscaled the production of gas diffusion electrodes, enabling more efficient microbial fuel cells and carbon dioxide electroreduction technologies for circular, low-carbon energy systems. A Fellow of the Royal Society of Chemistry, the Biotech Research Society, and the International Society for Microbial Electrochemistry and Technology, Deepak is also editor of Bioresource Technology Reports and the Journal of Environmental Chemical Engineering. Welcome, Deepak. The floor is yours.
Thank you so much, Anya, and good afternoon to all of you. A pleasure to be here and follow the three elegant speakers before me. So I will continue the story of microbial electrochemical technologies, and I will start directly with the upscaling part of this, or the efforts that have been made so far globally, by some of the authors of this lead article, but also by other researchers spread across the world. So what you see on the slide here is the first microbial fuel cell demonstration that was carried out already in 2007-2008 in Australia at the Queensland University researchers. What you see here is a tubular microbial fuel cells, 1,000-liter capacity, total 12 modules were built. And then what I want to highlight here is the real research in this field started in year 2000, and only after 7-8 years, what you see is a pilot that was operated on a real location. Now, in terms of technology readiness level, or TRLs as they are called, you can imagine this to be at TRL 7. And it's quite challenging, but also quite ambitious to go to this level already after 7-8 decade of fundamental research, because the field is quite interdisciplinary, as you might have guessed by now. It's a combination of so many disciplines here. You need microbiology, you need material science, you need to understand electrochemistry, and of course, engineering is a very big part of it. And therefore, this pilot was a good example of how all these disciplines come together. So moving on from here, then there were some other efforts. For example, this one I show is coming from Harbin Institute of Technology, HIT in China, where they already built a 10-meter cube stacked reactor. So when you do scaling up, you can have two ways. Either you can make an individual cell too large just by increasing linearly the size of the electrodes and the membranes that are used, or you can stack them up as has been shown here. And the total volume here was 10,000 liters, and the single module was 250 liters capacity. And I was checking if this paper appeared already in 2013-14, so almost 12 years ago, and they used mine tilling water to use as a substrate here or a feedstock to treat and produce electricity. This is how it looks like in practice. So this was the reactor schematics, which has been provided by Professor Yujie Feng and Weihua Yu. So when I was talking about upscaling, there are several challenges, and also the performance is not linearly similar when you go from a lab scale setup, which you can see on this image. On the left side, I will follow the same direction as Falk used. So you see a very small cell of 1 to 10 centimeters square, and in the bottom is the reactor, the famous called Cube reactor, developed at Penn State University and used by a lot of researchers all over the globe. In the middle is the rectangular shape of it, a little bit upscaled, going up to 100 to 200 centimeters square. And finally is a upscaled microbial fuel cell with using a meter square electrode. So the electrodes that are shown on the top, these are typical gas diffusion electrodes with the white part showing the gas diffusion layer, which allows the air or containing oxygen, because oxygen acts as the electron acceptor, to go into the electrolyte, and the other side is exposed to the electrolyte. So this project was done together with Professor Bruce Logan, who is also one of the co-authors of this lead article, and was tested with real human urine as substrate. And it was run for more than 6 months by them in a project that they did for US Army at that point of time. So it's very important to not only increase the size of the electrodes, but it also needs the reactor, corresponding reactor where you need to fit them, but also the membranes, because a lot of these reactors also use membrane or separators to separate the anode from the cathode part. The similar example, the second example, using the similar upscaled gas diffusion electrodes come from Ruhr University Bochum, where they used to treat 255-liter MFC module for municipal wastewater, real municipal wastewater. In the middle, you see stainless steel brush anodes. And on both sides of the anode, 2 cathodes were used to complete the circuit. And this experiment was also run for quite some months, showing the real field scale demonstration of a microbial fuel cell with a real municipal wastewater. In this slide, and this is also a part of the manuscript or the paper as well, which shows some efforts starting from the first one in 2008. This picture has been already shown. And the efforts that are spread all over the decade and the further after. So the first MEC pilot, the electrolysis cell to produce hydrogen, was carried out in Napa Valley in California. And this is the white reactor that you see, the 1,000-liter was operated at that time. Then in between, there were some 32 cell stacks MFC pilot, and the largest was actually an 850-liter reactor that was operated in 2022 with the 11 meters square of anode surface area and 20 meters square of cathodes. And currently, there are some large-scale METs are running in Spain, the so-called microbial electrochemical technology wetlands for treating the nutrient recovery as well as wastewater treatment. Moving further, I now present to you some commercial examples that have started in this field, which is also a quite nice efforts that were done. So the first example is coming from Cambrian Innovation, which was a spin-off from a university in United States. It was founded already in 2006. So this company was one of the first commercial examples that tried to incorporate microbial electrochemical technology in their operations. So what you see here, they started selling these containerized systems for wastewater treatment, but also biogas upgrading. And a part of it was linked to microbial electrochemical technology. And I will not go through the details, but you can see that a typical installation usually generated 30 to 200 kilowatts of power, and the BOD removal efficiency was quite nice, more than 85%. So they already targeted a couple of industries, especially wineries, breweries, and food industries were their first clients. The second example is called EMFCY, and this was originated in Israel. And they came up with this concept of electrobiogenic reactor. And afterwards, they shifted a lot of their operations in China. And recently, they were acquired by another company. So now I think they are known as Fluence, if you look them up. So EMFC, you will not find them by this name. but they are still in business, and a part of it is based on this electrobiogenic reactor, especially in China and a lot of Southeast Asian countries. They have systems that are up and running. And they claim at that time, and I made this slide a while ago, so they said the payback time for their systems was 5 years. But it's not only the energy recovery, but also the treatment part of it has to be considered here. The third example is coming from the neighboring country. So I live in Belgium, so this is coming from Netherlands, and this is a company called Planty, which was established already in 2009 and 10 as a spin-off from Wageningen University. And what you see here are microbial fuel cells based on a plant. So the root of the plant carries those electroactive bacteria and act as anode. And when you couple them with an air cathode, you complete the circuit and you produce some electricity. The electricity they generate is just enough to power the LED lights, as you see here, which are lighting a bike path there, but they also use it as to power the remote sensors. So they have several projects running in, for instance, in Indonesia. They have also demonstrated this in the rice fields. There, it's the whole field act as a plant microbial ecosystem. And they are still developing this system further for coupling it with home installations as well. Then the last example I give you is coming from my own organization, where we developed this idea of NDCOS, which is a trademark for anaerobic coupled digestion. And the idea was to upscale or up-concentrate the sewage with organic waste streams. And then in the first step, we used dark fermentation to produce biohydrogen. The effluent of this biohydrogen contains volatile fatty acids, which is an ideal substrate for the bacteria which are used in microbial fuel cells. And after several years of development, we demonstrated this on site in India, in south of India, for a canteen, which was producing food for 15,000 employees in a day. Afterwards, a second installation was also run in Vietnam. And the numbers you can read on the slide in terms of the sewage flow and how much biogas and electricity was produced using this system. And then this is really the last example that I would like to show. And this comes as a P-Power installation in Durban, South Africa. This was a project done by Ioannis Aeropoulos with the support of Bill and Melinda Gates Foundation. What you see here is a system, a stacked system, which produces electricity from human urine. And the light that you see glowing in the picture is used to charge the battery. And in the evening, the lights glows up the toilets. So it makes the atmosphere safe for off-grid installations, especially in rural villages where the grid electricity is not so easily available. So this was a real life installation and example that immediately had a societal impact as well. And there are several other examples which I have not covered because of the paucity of time right now, but you can find that more in the article from and the subsequent attachments. Now, I have shown you several examples where the MEC and MECs can be integrated in the whole treatment setups. I also saw some questions appearing while the previous speakers were talking about it, whether the energy use production is the main use of microbial fuel cells. But as you can see, there are several opportunities for METs to be integrated into the whole value chain. And these different colors indicates different places where you can say. So if you see a green dot, it means there you can have an application for MFC or MEC, either for energy recovery or chemical recovery. You can also have as a sensor, that's a dark purple one, or you can have a red dot where you can couple it with anaerobic digestion or for sludge conditioning and for high concentration liquids. So there are enough opportunities, and some of them needs to be developed further. Some are already ready for upscaling. And with this, I hand over the word to Uwe to conclude the presentation part.
Right. Thanks, Deepak. Yes, as a wrap-up, I would say we have seen quite some impressive development in the field over the past 25 years, and a field that is nowadays also covered in most of its part by a small society, an international society for microbial electrochemistry and technology. I think that was mentioned once already during the presentation. Now, it's really interesting to see that we have first commercial applications that start growing, although they are still, I would say, on a niche level. So it means that further work is certainly necessary when it comes to reactors, materials, to scalability, performance, durability, and also to cost efficiency. And that is quite a multidisciplinary task where we need material science, electrochemistry, biology, civil engineering, and many more science and engineering fields. It also means that we need also, or we have requirements on the legislative side where we have to make sure that we achieve the transition from the current linear economy, the linear wastewater economy, where we treat wastewater simply as a waste, to a circular economy where we use wastewater as a resource, and not just the wastewater, but also the ability to use the products like the nitrogen, like ammonia, phosphate as a product. I think that is not just a scientific, but also a legislative requirement. What we have to understand is when we look at individual aspects, it seems we produce only a little amount of electricity, but the nitrogen recovery alone might not cover the cost of the technology. What is important is we have a whole package with like a micro fuel cell and micro electrolyzer cells. We can achieve multiple things at the same time. We can clean the wastewater, reduce the cost for the energy, the energy cost for the wastewater treatment, and even be able to, on the long term, recover energy from the wastewater plus chemicals. So I think this is the package that makes MET so attractive. I think that was it from my side. I hope that the audience found our presentation useful, and I'm very curious to have a good discussion.
Thank you, Uwe, and thank you, Deepak. It was fantastic to see those pilot cases around the globe. Now I'm delighted to welcome our guest panelist and author of the Policy Outlook on the lead article, Professor Bruce Rittman, Centre Director and Professor at the Biodesign Sveta Center for Environmental Biotechnology at Arizona State University, USA. Bruce is Regents Professor of Environmental Engineering at Arizona State University and a pioneer in environmental biotechnology. His research focuses on the science and engineering of managing microbial communities to deliver critical services to society, including renewable energy generation, water and soil remediation, and improvements in human health. Author of over 850 publications, he co-authored the foundational textbook Environmental Biotechnology, Principles and Applications, and holds 21 patents, and has been recognized with major honors, including the Stockholm Water Prize and the Clark Prize for Outstanding Achievements in Water, Science and Technology. A very warm welcome to you, Bruce. Thank you for joining us. Now, let me start with a question to our joining panelists. So over to you first, Bruce. Your editorial argues that MET's earliest and most impactful role will be augmenting existing infrastructure, boosting digester performance, treating high-strength side streams, enabling real-time process control, rather than replacing conventional treatment systems. Having just heard the four authors lay out the full scope of what METs can achieve, does that pragmatic thesis still hold? And if augmentation really is the near-term opportunity, are the field's research priorities and investment structures currently aligned with that reality? Or are we still collectively chasing a more revolutionary vision that the data doesn't yet support? Your thoughts, Bruce?
Okay, thank you, Anya, for the introduction and that very provocative question. I'd also like to thank Uwe and Frontiers for inviting me to participate in this wonderful enterprise on MET. So in your introduction, Anya, you said, oh, I'm a pioneer. And it's true, I've been working in this MET business for about 25 years. And over those 25 years, I've noticed a couple of things. Number one, we have made tremendous scientific process at understanding the fundamentals, things like extracellular electron transport, materials, how to control the system is really, really impressive. In parallel, we've made some progress on creating technologically viable systems, things that could eventually get out in the marketplace. But what we've learned is that these systems are complicated, and there are a lot of technical challenges, engineering and materials challenges. And that leads me to my main response to your question here. These systems are pretty complicated. They have a lot of parts. They've got electrodes, membranes, circuits, control systems, and more. They're not cheap. These are going to be expensive systems. And therefore, in order to make them attractive in the market, we have to make the cost benefit work out well. And there are 3 points on how to do that. Number one is you want to produce value that has more economic value than just electricity. Electricity is cheap. And so you want to use those electrons that make it over to the cathode to produce things that are of higher value than electricity. And we've seen examples of hydrogen gas. Today, you can produce hydrogen peroxide, you can produce organics, that's so-called organic biosynthesis at the cathode. Those are all very much more economically attractive than to produce electricity. The second thing is that you want to get the most bang out of your buck from the capital investment. And one way to do that is to treat much higher organic strength streams. And I'm talking about things like from animal feedlots and dairies like that, food waste, food processing wastewater. You get so much more COD, and there are so much more of whatever the output is that it's going to really improve the financial situation a lot on that. And the third thing is, when possible, you want to leverage other investments that are already there. And so, for one example that was given by Deepak is integrating in with, say, sludge, anaerobic digestion sludge. And just to make the digester work a lot better so it produces more methane, it can take in more COD on that. And then you're leveraging the capital investment already made of anaerobic digester. So those are the key things that I think we need to do to make the METs really attractive on the market and not just exciting scientific area.
Thank you, Bruce. Now going to our questions from the audience. Andrei Kovalev, has the effect of direct interspecies electron transfer on microbial lifespan, including whether it increases or decreases growth and death rates, been studied? That's to you, Falk. And also a little addition to that, what do you think about the use of bacteriophages in microbial systems?
Thank you, Anya. So first of all, let's answer the second question. And related to this also, let's say maybe there was another question on fungi, if I'm not mistaken, in the chat. So there were already different yeasts shown to be electric, although the mechanism behind is quite, let's say, not understood yet because of the thicker cell wall and the glycan and so on and so forth. And for the bacteriophages, it was shown that they can selectively reorganize the EET facilitating proteins like OMCH in Shevanella, for instance, but the exact role is not really deciphered yet. So there are early indications, but a lot of things to study and a lot to understand. But now to your first questions, and I tried to read it in parallel, so I couldn't get the full question. So maybe you can repeat me or tell me who asked the question. The first
Andrei Kovalev asked, has the effect of direct interspecies electron transfer on microbial lifespan, including whether it increases or decreases growth and death rates, been studied?
Okay. So, I'm trying to recall. So first of all, we have to be sure that, or have to acknowledge that as Uwe introduced, the microorganisms need to do extracellular electron transfer to strive and to replicate and to be active. So there are some studies that also try to see for different potentials, different currents, how the life and death cycles of the bacteria changes. But one has to be clear that often these are done by dyes, like DAPI or Cyto, something like, or cytogreen. And the problem with these ones is that they sense a membrane potential. So what they sense actually are intact cells. They are often referred to as live and dead staining. But to my perception, this holds not true for electroactive microorganisms, because very often it's not clear if you get false positives or false negative results. And also what was introduced, I think by Deepak also, that we have a lower sludge production when integrating microbial electrochemical technologies with wastewater treatment, also shows that the biomass formation by the same amount of CO2 removal is lower, which is quite clear because part of this chemical energy goes to the microorganisms and the main share goes to the microbial electrochemical systems, because the sludge that we see in a technical system are merely the dead corpse of the microbes that we have that are there. So I hope this answers your question, but I'm happy to have my fellows adding here.
I might add, it's really interesting because we seem to understand a lot when it comes to single species, like model organisms like Geobacter, how they transfer electrons and how EET works. And then also we have the side that we know that we can actually treat complex organic solutions with the help of electroactive bacteria. But what we don't fully understand is actually the food webs that we have at the electrodes, how exactly basically certain compounds are broken down, and then basically the food is digested along that cascade, and electrons are transferred from one organism to another one. This kind of complexity is really difficult to study, and that is also one of the aspect that requires fundamental research. So not just development, but also understanding of this kind of complex biology.
Thank you both. Now, Isha Shakur has a question for Bruce. How do variations in microbial community composition influence the efficiency and stability of microbial electrochemical systems? And what strategies can be used to optimize these communities for consistent large-scale performance?
Yeah, wow. Well, that's a great fundamental question. That sounds like about 10 years of research. I mean, first of all, unless you're feeding something like acetate to the anode, you're going to get a community there. And even if you just feed acetate, you probably will get a community anyway. So the key here is, when you have these other organisms who are not electrogenic, how do they interact? And we want them to produce, the organic acids that the electrogenic bacteria are going to use. But we don't want them to physically squeeze out the electrogenic organisms either. So this is, a really fundamental challenge. Now, I do want to say that one of our control knobs, things we can do, is controlling the anode potential. And that's one of the unique things about these microbial electrochemical systems is you can control the anode potential, and it does a couple of things. But the thing that's really valuable is that it allows you to tune how much energy the electrogenic bacteria are going to harvest for themselves versus how much energy on the electrons that leave. And so you need to poise this just right. You got to give them enough energy that has the potential negative enough so that they get enough energy to grow. But you don't want to make it too high, because then you've got activated sludge and you're producing all this biomass. So the one of the things we have learned is for the electrogenic bacteria is you really have to control that anode potential very carefully on it. Now, if you do it, you poise it right, at least you get them in the biofilm, and they'll probably be closest to the anode because they respire to the anode. And the other organisms are more likely to be towards the outside of the biofilm and delivering things like acetate to them. Now, the other factor, of course, is a detachment, which we don't understand very well in these microbial electrochemical systems. But assuming that the fermenters are out near the outside, they're going to be the ones that are going to be subject to detachment. And again, you want just the right amount of detachment so that you don't overgrow the biofilm. But on the other hand, if you have too much detachment, you don't get the fermenters. So this is a really fundamental research area, but it has to be integrated with practical systems because the physical environment, it's the physical environment that controls detachment. And so this has to be studied at a pretty large scale to get anything realistic.
Thank you, Bruce. Now, Falk, we have a question from our audience in the China stream. In practical applications, how can the purity and safety of recovered products such as struvite or ammonium salts be ensured so they can be safely returned to agricultural land as fertilizers, thereby closing the resource loop from toilet to farmland?
This is a very interesting question, I have to say. When we come to products, and the struvite was mentioned, first I would like to go to ammonia and ammonium, because here we have the advantage that we have a transfer via the gas phase, as I've introduced, and therefore there is the risk of cross-contamination is not zero, but it's certainly lower than when you go for struvite, where you have sulfate precipitates from urine that are used there. And there are studies that also shown that metabolites of drugs and as well of other substances that are in the urine and that are excreted by humans are still to be found attached to this, let's say, to the product. And it certainly will need some, let's say, purification and also some industrial technical checks to show that this has really that this is safe and sound to reuse them. And I'm not aware of research that so far went all the way down from, let's say, real using urine with microbial electrochemical technologies, recovering a product and making all, let's say, the legislative steps that are needed. But there are tests that already have shown that you can grow plants with this. And I'm happy if one of the fellows wants to add here.
Does anyone want to add, or I'll move on?
Just to add slightly to that, is that the main job of the microbial fuel cell is to kind of concentrate up those resources. So we're not going to get them to a pure state, but we will be able to concentrate them up or down so that then it's easier to chemically extract and treat and put them into a reuse situation.
Liz, there's a question for you as well from Pablo Alcalde. Could this work in large refugee camps as an alternative to classical desludging management?
That's a challenging question and a challenging place to kind of put a new technology in. As Bruce alluded to, these are really difficult, complicated technologies to actually operate and to use. So yes, it possibly could. Whether that is the best place to use it and to start deployment, I would argue not. I think it's in that situation, you would want to do something simple, basic, and safe that gets the job done with as least risk as possible. I think, as Bruce mentioned, METs have got better places somewhere else. In my opinion, others may think differently.
Thank you, Liz. Now, Uwe, there's another question from the China stream. What are the most promising engineering strategies currently available to address scalability bottlenecks in microbial electrochemical technologies, such as increased internal resistance and reduced performance during scaling up? What are the most critical technical hurdles that must still be overcome before MET reactors can become a standard component of municipal wastewater treatment plants?
All right. Yes, but it's interesting question, really important one, because we have to understand that this is we have some contradiction here. On the one hand, we have to treat large volumes of wastewater, and this is why we have to scale things up. And on the other hand, we have an electrochemical technology. And electrochemistry, we need a current to flow, for which we need, for efficient cells, we need electrodes that are close to each other in order to avoid ohmic and thus energy losses in the cell. So scaling up means if we would simply scale things up in a way that, like, also in order to have more water to flow through, that the distance between electrodes becomes larger, the electrodes themselves become larger. That means that runs into inefficiency. So one of the challenges is, do we scale up or do we number up cells? Meaning, do we rely? It's like the cells in our body. Like biology didn't choose to compose us from, I don't know, 3 single big cells, but chose to use small cells that are more efficient with all the aspects, mass transport, and so on. Now that makes things again more complicated, because when you have more individual cells, that might cause other problems. I don't have one single answer. That is one of the complicated questions in the field. We need materials, electrodes that at larger scale have a high enough conductivity. That was one of the problems. Deepak showed the first pilot plate from 2006 from Queensland. Cornell Rabe presented that. We had long electrodes which had a too high ohmic resistance that basically caused ohmic losses. So that is the challenge, to have materials that are cheap enough but still have good enough electric conductivity, for instance. We need also reactor designs that allow us to scale or to number up without losing too much efficiency. There are different opportunities, but it's not like simple to explain exactly how to do that. It's really a tricky interplay of phenomena.
Thank you. Now to Deepak from Mary Kruger. What do you see as the biggest operational implementation challenges for this technology into established wastewater treatment plants and water resource recovery facilities?
Thank you. I was thinking, because I have been handling a lot of questions online, I have already answered 9. But I wanted to give a word before I answer this question to the people I answered online. If they have a follow-up or a rebuttal to my answer, please, you can still type there, and Anya will try to get it back to me, or the fellow panelists. If they can have things to add there, please feel free, though I'm trying to address them as good as possible. Now, coming back to question from Marie, which is indeed the first one now on my list. So where to start, actually? So there are many challenges. This has been enumerated not only in the lead article, but also by ourselves today by different speakers. The main thing is at 2 different levels, if I can put it this way. One is the technological, the other one is related to the policy level as well, so to speak. Now, we talked about, for example, nutrient recovery, or anything that you make out of microbial electrochemical technology, whether it's a single cell protein for food or feed applications, or a nutrient that somehow enters your food cycle. There is immediately some kind of, so currently we make, for example, nitrogen-based fertilizers with Haber-Bosch, apply in the field, and you forget about it. And no matter what the nitrate or nitrogen pollution comes, it's then somebody else's problem. Whereas if somebody proposes an MET-based approach, so there will be already so many checks and balances that you have to prove it's safe for humans, safe for animals, of course. So that kind of policy support is needed. The second big challenge is, of course, the cost and the cost of the materials. In 2008, there was already a paper in Trends in Biotechnology. And if I remember those numbers correctly, it was like 1,000 times more expensive, or 10,000 times more expensive compared to anaerobic digestion, to cost of treating 1 kilogram of COD. Now, in these intervening 15, 20 years, the cost has gone down a lot. A lot of new, cheaper materials have been developed, but we are still not there. And then I just answered a follow-up question or link to it, that why should somebody choose a MET-based treatment approach over a conventional? But there is no competition as we see it, at least from my side. I see them as complementary. It's either a pre-treatment or a post-treatment where METs should be integrated. So that's how I see it at the moment.
Thank you, Deepak. Now, one for Bruce, maybe Liz, you might have a few comments as well. Ashif Yadav writes, as we all know, the continuous redefinition of goals may dilute progress toward deployment. Do you think the field needs to move beyond incremental studies and stabilize its core objectives to achieve meaningful technological advancement? Or are microbial electrochemical technologies and bioelectrochemical systems better regarded as tools for addressing specific scientific questions rather than practical technologies? Bruce, perhaps you have some thoughts.
Got it. Okay, unmute. Yeah, that's really a great question. The second part is, about the microbial electrochemical systems as research tools, and they are research tools. They are very powerful research tools. We've been using them. We've been gaining a lot of knowledge about many things, about microbial ecology and physiology and things like that. And we should continue to do that, and we will continue to do that. But that's different from, producing a scaled up commercial technology. My personal opinion is that we should shift our focus away from treating domestic wastewater. I don't think that's going to ever work. We can improve anaerobic digestion. We can treat high strength wastewaters. I think those are where we ought to put our focus now.
Liz, have you got any further thoughts?
Agreed. Yeah, well, I'm not going to disagree with Bruce Rittman. But yeah, I think they're two very separate paths, and maybe this is the point at which this field diverges, that, the technology works at a basic level and put in the right place in a treatment train where you've got high strength wastes that are difficult to deal with any other means. And if you can treat them effectively and at the right cost with an MET, that's great. That technology can get out, and I think will get out into the commercial world. But it is a fascinating tool, and I think research will continue for a long time in this area, because the ability of a microbe to produce an electron as it's eating in real time, and you can see that on a computer, means that we can quite literally look into the world of bacteria and see it metabolizing in front of our eyes. And I think that will fascinate research for a long time to come, and result in many good discoveries.
Thank you, Liz. Now, Uwe, Sharon Velasquez writes, what is the most challenging issue affecting process efficiency and scalability of MET systems?
I think it's pretty much what we discussed before. It's efficiency loss due to increasing resistance, ohmic resistance mainly. I think that is the main one main point. I think there's also connected to another question. In a lab, we very often work with not just with single strength, but also with easy to digest substrates. In reality, we have more complex substrates, also substrates that is fermentable, which is a requirement for many electroactive bacteria to digest that substrate. But that also limits the efficiency of the process, like the so-called Coulomb efficiency or Faraday efficiency. So the number of electrons gathered in the electrochemical process, well, in relation to the COD removal or the removal of organics. But that is something that we don't actually have in our hand. What we do have in our hand is the reactor design that allows us to reduce these electrical losses, and having then more like less losses means also you have more efficient electrode potentials and most likely or more efficient bioelectrochemical substrate conversion. So that's one point that also impacts the other.
Thank you. Now, this is an open question, so whoever wants to take it up, please do. And Diana Rai asks, what are the key technical benchmarks and performance indicators needed for the successful scaling up and commercialization of microbial fuel cell technology in wastewater treatment? Any takers?
I can have a first go at this, and then maybe others will add. One of the critical points that sometimes is overlooked is the treatment rate per surface area of land. Urbanization is where most of the population is growing, which means we're getting a mass of people producing massive wastewater in urbanized sites. So we need the technology to be able to treat high concentrated wastes wherever they occur, but do so in a small footprint of space. Because if we had a huge amount of space, we can do it with a pond and a lagoon, which is way cheaper and simpler. So that will be my major factor. But Uwe is going to jump in with another one.
Yeah, there's some points that we don't have control over. And one of the points is to say that this is kind of external factors, because at the moment we say, what does it mean to be economic? That is always like, if we want to recover nitrogen or like ammonia and phosphorus, that means like, what is the cost of recovering these elements or these compounds in relation to the market price? And at the moment, it may not be cost efficient, but we have seen, like with the resources of phosphorus, the phosphorus price of the phosphate price has gone through the roof, and that's also a geopolitical or geo resource thingy. So we have to be prepared to at some point have a technology ready and being able to instead of, I mean, the phosphorus or phosphate resources are limited, and on the other hand, we have the ammonia, which when we want to produce it from the nitrogen from air costs a lot of energy. And when basically energy prices further go up and also phosphate prices go further up, that might be at some point also the level where the technology might jump in and become economically viable. And that's why it is difficult to actually say we have this benchmark, we need this and that watts per square meter or this or that efficiency value. This is always depending also on the primary goal of your treatment train, for instance, and of your recovery train.
Thank you, Uwe. Now, continuing more with the financial side, Deepak Samir Baipa is asking, one major challenge in scaling up these technological solutions, particularly in countries like India, is that the return on investment may not appear attractive. How can this challenge be overcome?
Yeah. So for now, it links to another question that I answered in the chat directly. So someone asked, when does this moves from research to commercial application? And for me, the moment any company starts making money, either by selling a product in the market or as a service, they are in business. So it's not only a real large scale treatment system, and this also links to the others. The sensors are already in use for many years now. As environmental sensors, the U.S. Naval Research Laboratory has been applying them for many years. The Plantee example that I showed you, their sensors are running in oceans and also in wetlands to monitor the rain and sunlight and these kind of things. I'm also in contact with a couple of small companies, startups, who are trying to develop some sort of this business in India. But at the moment, they are all in the exploratory phase. Indeed, they don't have a ready product to sell in the market. The best they can do is to find a niche and to couple it to a particular application area. And then only the example that I showed that we made was in a software company actually, but the canteen, it was producing a large amount of organic waste. They wanted an off-grid standalone treatment system, and it was not MET alone. It was always a treatment chain, with some pre-treatment, some post-treatment, membrane-based, anaerobic digestion, and then MET was taking care of a small part of it. So my advice will be to look into such scenarios where METs can play a tangible role, even with the performance that they have right now.
Now to Liz, looking at regulatory frameworks and market mechanism. Michael Hananto Utomo writes, how should researchers build effective stakeholder coalitions with industry and government to address economic viability and scale up microbial electrochemical technologies? What are the most important lessons from existing pilot deployments of MET-based wastewater treatment and nutrient recovery systems?
Gosh, it's a complicated question. So, I mean, how to build stakeholder engagement. The key thing is to be actually talking and engaging with stakeholders. Researchers are very good at going off and solving a problem that may actually not be a problem. So if you start those conversations early, you know what the pain points are of those industries that you're dealing with. You know what metrics are important to them and where they need to get and where a technology needs to sit to be viable within their company. Then you can tailor that research towards it, if that's the goal of the research. In terms of legislation, again, really difficult to kind of change legislative behaviour. You know, that's quite difficult to influence and to put into practice. But it's about getting people in the correct conversations. Not sure if anybody else wants to jump in there.
No, I totally agree with what you suggested, but also speak out. I mean, if you see a problem, if your community or your technology, you see there is a problem, you need to talk to, unless you make your voice heard. And this forum is actually providing an opportunity like this. So we often talk to, let's say, policy makers, the people who actually make these decisions. So, and when we talk to them, we realize they don't even realize that this type of research, or not take MET as an example, but any upcoming technology, half the time they are not even aware that such a problem exists. So unless you speak out, but speak out means not only loudly, but also write about it, post about it, and then someone somewhere will hear it. And once the problem is known, then of course there are ways to find a solution.
Thank you, Deepak. Now, Falk, Pedro Bartemes Filo asks, what would be the most effective use of low-cost wireless devices for detecting microbes and contaminants using electrochemical technologies?
This is thank you for the questions. Also, good questions. What is the most effective use? And it's again, what kind of efficiency do we mean? So as Deepak also stressed already, there is for the water and wastewater treatment, there are already sensors in the market when it comes to COD sensing and this kind of thing. So this is already available. But this, of course, is let's say, is a very specialized niche market. But we also can go to other niche markets, other applications, and these are quite diverse, because the electroactive microorganisms can be found finally everywhere, or the colleagues of us termed it in an article, are all microbes electroactive, so we can find them. Just to give you one idea is that we also have electroactive microbes in our gut, people are now considering that to have electroactive sensors in the gut or at the skin to monitor health conditions. I think this is still a little bit far-fetched, a little bit of nice visioning on dreaming, which I really like. But if we go into this kind of direction, this, of course, will be to open an entirely different market, and also a different market volume that can be touched with this kind of technology. And this also goes for bio-based electronics, where on electroactive microorganisms. So you can not only use it to make sensors, but also to create circuits and circuits boards and logic gates and all this kind of things. So bio-based computing maybe is on the same level of visioning. This is something also that we can see I would hope to see in the far future. And I think this would be, a total turnaround in terms of volumes and in money, as well as depth of penetration into our society.
Falk, thank you very much for that future gazing there. And unfortunately, we've run out of time for today's discussion, but I do have just enough time to ask all my panelists for their final takeaway message. What point reflection would you like to leave viewers with? Liz, could we start with you, please?
I was hoping you weren't going to say that, because I'd forgotten you were going to ask this question at the end. My takeaway point is that these are fascinating, exciting systems, and let's hope in the next 10 to 20 years they continue to explode in the way that they have done, and we learn lots more, and we actually see them deployed in real life.
Bruce.
Yes, thanks. So I've emphasized that we need to have a good value proposition. We need to harvest real value from these systems. I continue to say that. And I also think on the practical scale up, it's going to be modular system, and we're going to get economy of scale, not by making them bigger, but making a lot of them. And it's going to be really cheap to manufacture.
Falk, your final thoughts, please.
My final thoughts is on the fundamental side. I hope that with the future we will understand what the role this microorganisms in the early formation of our planet Earth have played. This is something we have a little bit touched today, but not too much. But at the same time, also explore how they will shape our future with different kinds of technologies, also on planet Earth and beyond.
Thank you. And Uwe, your final words, please.
Right. Right. I mean, I'm an electrochemist. I'm coming from the fundamental perspective, and I find that always really fascinating. It's a fascinating field, and I hope that we can achieve really interesting things. I see on a bigger picture, what we have to really learn is, as mankind, to um, not to leave our dirt behind simply as a waste. We need to get to a circular economy because simply we can't afford it anymore the way we actually um, we live. And that is something why we desperately need to develop technologies. And one technology could be the MAT as one technology, but it can also be other technologies. And that's why we need to develop these kind of tools for the future.
Thank you to everybody today, all our experts. That was a really fantastic session. To remind you, we had Dr. Uwe Schroeder, Dr. Liz Heydrich, Professor Falk Harnisch, Dr. Deepak Pant, and Professor Bruce Rittman, and they all joined us today. And again, thank you to you for all joining us today. I hope you enjoyed the session. Please do visit the Frontiers in Science website to read the article we've discussed today and its related content. You can continue the conversation on social media using the hashtag FrontiersIn. It's just appeared. So for now, from me and from our guests and the entire team at Frontiers, thank you experts and thank you for watching. Goodbye.